Hello, I'm Peter Newman, and this week's AHRI Insight we're featuring some research done by Jenna Malone at the University of Adelaide. Jenna works on the research team that's led by Chris Preston and is funded by GRDC. So what they were looking at was the mechanism of resistance for glyphosate in brome grass. So they had two populations of brome grass, one from South Australia, one from Victoria, about 400 kilometres apart, so they probably evolved separately, and they found both had the same resistance mechanism. The mechanism was gene amplification. So this is where the plant, instead of having one copy of the gene, in this case it had 20-fold number of copies of the gene. So the plant overproduces the enzyme, EPSPS, that glyphosate is trying to bind to. And so while glyphosate can still bind to this enzyme and inhibit some, there's still plenty left over for the plant to grow as normal. This is interesting because it's the same resistance mechanism that Palmer amaranth on the other side of the world in the USA is causing big problems. Gene amplification is the main resistance mechanism in Palmer amaranth, which is arguably the world's biggest herbicide resistance problem. So very interesting that two very different weeds on two different continents evolve the same resistance mechanism. For more information, have a look at AHRI Insight.